Hello friends, welcome to Unlocked Science by Mishtab. No cramming, just understanding. Every cell in your body traces its story back to one small beginning. From that single cell emerged the skin, nerve, and blood cells, each with a unique purpose. How did such beautiful complexity begin? The answer lies in two remarkable processes: cell division and cell specialization. By the end of this video, therefore, you'll be able to define cell division and identify the different types of cell division, differentiate the types of cell division, give the importance of cell division, define cell specialization, and describe how cells become specialized to perform specific functions. Let's dive in. Cell division is the biological process where a parent cell replicates and physically sprints to create two or more daughter cells. For single-celled organisms like bacteria, one single division equals to one new offspring. However, for multicellular organisms like humans, it is the process that turns a single fertilized egg into trillions of specialized cells. Types of cell division. There are three types of cell division, and the type of cell division depends on the type of the cell and the goal of division. We have binary fission, mitosis, and meiosis. Let's begin with binary fission. Binary fission is the simplest form of division used by bacteria and archaea. Those are prokaryotes. Because these organisms lack nucleus, the process is rapid and efficient. How it occurs? The single circular DNA molecule replicates, forming two DNA loops. The two DNA loops attach to the plasma membrane, and as the cell grows, they are pulled apart. The cell membrane then pinches inwards, creating two identical clones. It is incredibly fast in that some bacteria can divide every 20 minutes provided the conditions are favorable. Interphase. Before any other type of cell division, be it meiosis or mitosis occur, interphase occurs first. Interphase is often called the resting period because during this time, the cell is metabolically hyperactive. It is divided into three distinct stages, that is, G1 phase, the first gap, S phase, known as synthesis phase, and G2 phase, known as second gap. During G1 phase, the cell grows physically larger, copies organelles like mitochondria and ribosomes, and makes the molecular building blocks it will need later. The cell ensures that it's healthy enough and has enough resources to divide. If not, it may enter GO phase, where the cell can no longer divide. The S phase. The cell replicates its entire DNA. By the end of this phase, every chromosome consists of two identical sister chromatids, joined at a central point called centromere. The DNA count has doubled, but the chromosome count remains the same. In animal cells, the centrosome, that's the microtube organizing center, also duplicates to help the chromosomes pull apart. The G2 phase, that's the second gap. This is the final check and preparation period before the cell enters mitosis or meiosis. The cell undergoes a final burst of protein synthesis, specifically tubulin for spindle fibers, and accumulating ATP reserves to fuel the energy-intensive mitotic process needs. It performs a rigorous DNA proofreading, check to identify and repair any replication errors, ensuring that the genetic material is intact and accurate before the cell officially enters mitosis. After interphase, the cell can decide to enter either mitosis or meiosis depending on the goal of cell division. Let's dive into mitosis. Mitosis produces two genetically identical daughters from a single parent cell. These daughters resemble exactly the parent cell. This process is involved in body growth and repair. Stages of mitosis. There are 4 stages of mitosis, that is prophase, metaphase, anaphase, telophase. During this process, several things happen simultaneously. Condensation of chromosomes, where the DNA fibers coil tightly into visible chromosomes. The spindle fibers begin to form, where the microtubules, that's the spindle apparatus, begin to grow from structures called centrosomes. The nuclear envelope, that's the shell around the nucleus, dissolves along the spindle fibers to reach the chromosomes. The cell then enters into metaphase, which is the middle stage. During metaphase, the chromosomes, which consist of paired sister chromatids, align in a single file along the metaphase plate, that's the equator or the middle of the cell. The spindle fibers attach the kinetochore of each sister chromatids before proceeding, ensuring accurate division. The cell then enters anaphase. During this stage, the sister chromatids are pulled apart at the centromere by the spindle fibers. The now separated sister chromatids, of which they are considered to be individual chromosomes, move to the opposite poles of this cell with their centromere leading. The cell physically lengthens, preparing for cell division. Telophase. In telophase, the chromosomes reach the opposite poles of the cell and start decondensing back into chromatins. Nuclear envelope reforms around each set of chromosomes, creating 2 separate nuclei. The mitotic spindles break down and the cell then divides. And in animal cells, the cell membrane pinches in, creating cleavage furrow that separates the cytoplasm into 2 distinct daughter cells. While in plant cells, a cell plate forms in the middle, which develops separating the daughter cells. Beyond the intricate dance of chromosomes during its physical stages, mitosis serves as the fundamental mechanism for life's continuity and maintenance. Importance of mitosis: growth and repair. It allows a single fertilized egg to develop into a complex multicellular organism. Without it, organisms couldn't increase in size or complexity. Tissue repair and replacement. Mitosis is responsible for healing. When you get a cut or a break bone, your body uses mitosis to produce new identical cells to replace those that were damaged or have reached the end of their lifespan, like skin or blood cells. Asexual reproduction. For many single-celled organisms, mitosis is the primary method of reproduction, ensuring the population grows with genetically identical offsprings. While mitosis is the ultimate tool for biological consistency, it reaches a limit when it comes to the future of a species. To move from the survival of an individual to the creation of a new generation, the body switches gears from mitosis to meiosis, trading carbon copy stability for genetic variety. This drives us to meiosis. While mitosis produces identical body cells for growth and repair, there is another type of cell division that is equally important for reproduction. This process is known as meiosis. Meiosis, therefore, is a type of cell division that occurs in reproductive cells and produces 4 genetically different daughter cells, each containing half the number of chromosomes from the parent cell. These daughter cells are different from each other and from the parent cell. This process is essential for the formation of gametes, such as sperm cells in males and egg cells in females. The process occurs in reproductive organs, also called gonads. That is the testes in males that produce sperms and ovaries in females that produce egg cells. Understanding meiosis requires examining the key characteristics that distinguishes it from other forms of cell division. Characteristics of meiosis. Meiosis occurs in germ cells found in reproductive organs. It involves two consecutive divisions, that is, meiosis 1 and meiosis 2. It produces 4 daughter cells. The daughter cells are genetically different from each other and from the parent cell. Each daughter cell contains half the number of chromosomes of the parent cell. These characteristics arise because meiosis occurs through successive divisions known as meiosis 1 and meiosis 2. Meiosis 1 targets at reducing the number of chromosomes, where meiosis 2 is similar to mitosis. The first of these divisions, that is meiosis 1, plays a crucial role because it reduces the number of chromosomes by half, and therefore it is often called reduction division because homologous chromosomes are separated, reducing the chromosome number from diploid, that is, to n haploid, that is n. Stages of meiosis 1. Just like other types of cell division, meiosis 1 progresses through a series of well-defined stages, that is, prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1. Meiosis 1 begins with prophase 1, the longest and most complex stage of meiosis. During this stage, several things occur, among which is liptotin. In liptotin, chromatin begins to condense into visible thread-like structures. Each chromosome consists of 2 sister chromatids, zygotin. Homologous chromosomes that are matching in pairs from each parent find each other and begin to pair up in a process called synapsis. They are held together by a protein structure called the synaptonomal complex. Packaging. The pairs are fully joined, forming a tetran, that's 4 chromatids. This is when crossing over occurs, where non-sister chromatids physically break and exchange segments of DNA. Diplotin. The synaptonomal complex dissolves and the homologous chromosomes begin to pull apart. However, they remain attached at specific X-shaped points called chiasmata, which mark where crossing over happened. And therefore, the product of diplotin is genetic recombinance, diakinesis. The chromosomes reach their maximum condensation, the nucleolus disappear, the nuclear envelope breaks down, and the meiotic spindle begins to form. The cell then enters metaphase. During metaphase 1, one important feature that occurs is that homologous chromosome pairs are aligned randomly at the equator. This means that the maternal chromosome or the paternal chromosome can face either of the pole of the cell. That's they randomly mix. And they do that in a double row at the metaphase plate. Mathematically, with 23 pairs in humans, they are 2 power 23. That's over 8 million possible ways through which these pairs can align themselves at the equator. This is why you don't look exactly like your siblings. Because of this random alignment, the chromosomes that move into the daughter cells will be of different combinations of maternal and paternal chromosomes. Once the homologous chromosomes are properly aligned at the equator, the cell proceeds to the next stage, known as anaphase 1, where these chromosomes begin to separate. During anaphase 1, the homologous chromosome pairs that were aligned at the equator begin to separate. Each chromosome in a pair moves towards the opposite poles. It is important to note that the homologous chromosomes separate, but the sister chromatids remain attached towards the centromere. Simply, we are separating chromosomes, not chromatids. The spindle fibers shorten and pull the chromosomes towards the opposite poles so that each pole receives one chromosome from each homologous pair. As a result, each feature daughter cell receives only one chromosome from each homologous pair. The physical act of these pairs moving apart is called disjunction. If this fails, it is called non-disjunction, which leads to an extra or missing chromosome in the offspring, and that's Downey's syndrome. Since chromosome pairs were randomly arranged during metaphase 1, the chromosomes that move to each pole contain different combinations of paternal and maternal genetic material. This contributes to genetic diversity in offsprings. Telophase 1. Following the separation of homologous chromosomes, during anaphase 1, the cell enters the final stage of first meiotic division known as telophase 1. And in this stage, the homologous chromosomes that separated in anaphase 1 arrive at the opposite poles of the cell. Each pole now contains one chromosome from each homologous pair, indicating that each pole is haploid containing half the original number of chromosomes. In many cells, a nuclear membrane begins to form, enclosing the set of haploid chromosomes. However, in some organisms, the nuclear membrane may not fully form at this stage, cytokinesis. After the chromosomes reach the opposite poles, the cytoplasm of the cell divides, a process known as cytokinesis. This division results into formation of two daughter cells, each containing haploid number of chromosomes. Although the chromosome number has already been reduced during meiosis 1, the process of meiosis is not yet complete. Each of the two haploid daughter cells therefore undergoes a second division known as meiosis 2. Unlike the normal cell cycle, a complete interphase does not occur between meiosis 1 and meiosis 2. This is because the DNA replication already happened before meiosis 1, and the chromosome number has already duplicated. Instead, the cell may go through a short resting period called interkinesis, during which the cell prepares the second division without replicating DNA. This is important because if DNA replicated again, the chromosome number would double, defeating the purpose of meiosis. Meiosis ensures that the final gametes are haploid, ready for sexual reproduction. Stages of meiosis 2. Meiosis 2 closely resembles mitosis because it involves the separation of sister chromatids through a similar sequence of stages. They are 4 stages: prophase 2, metaphase 2, anaphase 2, and telophase 2. Prophase 2. During this stage, the nuclear envelope, if they formed in the telophase 1 of meiosis 1, break down again. New spindle fibers form and begin to grab the chromosomes. Unlike prophase 1, there is no crossing over here. The gene mixing is already done, and now it's just about organization. The chromosomes we have here are recombinants. Metaphase 2. The chromosomes align themselves at the metaphase plate in a single row, exactly like they do in mitosis. While they are in a line, the sister chromatids are no longer identical anymore because the crossing over that happened in meiosis 1. Anaphase 2. The centromere finally breaks and the sister chromatids are pulled apart towards the opposite poles, with their centromere leading. Once they are separated, they are no longer called sister chromatids, but now as individual chromosomes. Telophase 2. Nuclear membrane form around the 4 sets of DNA. The outcome is that the 2 cells of meiosis 1 divide again, resulting into 4 daughter cells in total. These cells are haploid, that each contains half the original number of the parent cell, and genetically unique due to crossing over and independent assortment. None of the four cells are the same as each other or the parent. And that's how you can go from one blueprint to four unique possibilities. In males, the four cells become sperms, and in females, one become a large egg, that's the ovum, while the others are polar bodies. Meiosis is the reason why life is a constant shuffle and never repeating the same hand twice. Although meiosis 1 and meiosis 2 occur consecutively during meiosis, they differ in several different ways. And this drives us to differences between meiosis 1 and meiosis 2. Meiosis 1 is also known as reduction division because the chromosome number is halved, while meiosis 2 is called equivalent division because the number of chromosomes remains the same. Meiosis 1 reduces the cell from diploid to haploid, while meiosis 2 maintains the cell haploid. Meiosis 1 aims at separating homologous chromosomes, while meiosis 2 separates sister chromatids. Crossing over and independent assortment occurs in meiosis 1, while no crossing over occurs in meiosis 2. DNA replication occurs before meiosis 1, while there is no DNA replication before meiosis 2. Meiosis 1 gives us two haploid daughter cells with X-shaped chromosomes, while meiosis 2 gives us four haploid daughter cells with I-shaped chromosomes. Similarities between meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2 are consecutive divisions that make the process of meiosis. Each division proceeds with four main stages, that is prophase, metaphase, anaphase, and telophase. Spindle fibers form and help chromosomes move and align themselves at the equator, and later pulled to the opposite poles of the cell. And both of these divisions end in cytokinesis, which involves separating the cytoplasm, producing new cells. Having examined the two divisions of meiosis, it is important to understand how meiosis differs from mitosis, another important type of cell division. Differences between meiosis and mitosis. Mitosis involves one division, while meiosis involves two divisions. Mitosis results into two identical diploid daughter cells, while meiosis results into four haploid daughter cells that are genetically different from each other and from the parent cell. In mitosis, homologous chromosomes do not pair, while they do pair in meiosis, particularly in prophase 1. Crossing over occurs in meiosis, while it doesn't occur in mitosis. Functionally, mitosis is important for growth, repair, and maintenance, while meiosis is important for production of gametes for sexual reproduction. This difference becomes clearer when we consider what would happen if these processes were reversed, that if meiosis is involved in growth and repair and maintenance, and mitosis is involved in production of gametes for sexual reproduction. If your skin cells used meiosis, you could have a different face every time you healed from a scratch, because meiosis produces genetically different cells. Conversely, if reproductive cells divided by mitosis, every child would be genetically identical to the parent. There would be no variation. Mitosis and meiosis share several fundamental features as processes of cell division. This drives us to similarities between mitosis and meiosis. Both are forms of cell division. Both processes involve the division of a parent to produce new daughter cells. Both begin after DNA replication. In both meiosis and mitosis, DNA replication occurs during interphase, before the division begins. Both involve similar stages, that's prophase, metaphase, anaphase, and telophase. Both involve spindle fibers. that help separate chromosomes and move them to the opposite poles. Both ensure genetic material is distributed into the daughter cells. In both, chromosomes are carefully separated so that each daughter cell receives genetic material. Both end with cytokinesis. In both cases, cytoplasm divides to form separate cells. Cell division increases the number of cells in an organism, but for a multicellular organism to function efficiently, these newly formed cells must acquire specific structures and functions. This transformation of identical cells into cells with unique functions is known as cell specialization. Let's dive into cell specialization. Cell specialization, also called cell differentiation, is the process by which stem cells develop specific structures and functions that allow them to perform particular roles in the body. How possible is this? Cell specialization occurs in several ways, among which is gene expression. During development, every cell except red blood cells contain your entire genome, but not all genes in a cell are active at the same time. During differentiation, some genes are turned on, that's they are expressed, and others are turned off, that's they are suppressed. For example, a skin cell has the instructions to make insulin, but these genes are turned off. Similarly, a pancreatic cell has instructions to grow hair, but these genes are also switched off. This selective expression causes cells to produce different proteins, which leads to different cell structures and functions. Another way in which cells become specialized is through cytoplasmic determinants. This happens during the very first division after fertilization. An even distribution in which the mother's egg cell isn't a perfect soup. It has different proteins and RNA scattered in different areas. And when the cell divides, one daughter cell might get more of protein A, while the other cell gets more of protein B. This immediately sets them on different developmental paths and thus specialized. Cell signaling and induction is another way how cells become specialized. Cells talk to their neighbors to decide what to become. That's the neighborhood effect. A cell's fate is often determined by its location. And induction, whereby nearby cells release chemical signals called morphogenes. If a stem cell receives a high concentration of a heart signal, it begins changing its structure to become a cardiac muscle cell. Those are the three ways how cells become specialized. Let's look at how do you identify a specialized cell, and that's the characteristics of specialized cells. Unique morphology. Their physical structure is specifically designed for their task. For example, red blood cells are biconcave to maximize surface area for oxygen. Sperm cells have flagellum for swimming. Specific organelle density. They often have more of specific organelle than a standard cell. For example, muscle cells are packed with mitochondria for energy. Pancreatic cells have extra ribosomes and endoplasmic reticulum to produce digestive enzymes. Surface area adaptations. Many specialized cells have folds or extensions to increase absorption or communication. For example, root hair cells in plants have long projections to soak up water. Neurons have long axons to send signals over distances. Specialized proteins. These cells contain unique proteins that other cells don't. For example, a muscle cell contains actin and myosin for contraction. Red blood cells contain hemoglobin. Loss of certain functions. To become highly efficient, some cells lose standard parts. A good example is the red blood cells. They inject their nucleus to make more room for oxygen. Importance of cell specialization. Division of labor. Cell specialization allows different cells to perform different functions within an organism. Instead of all cells doing the same task, each cell type focuses on one specific role, making biological processes more efficient. For example, red blood cells transport oxygen, nerve cells transport signals, muscle cells produce movement. This distribution of tasks is called division of labor. Formation of tissues, organs, and organ system. Specialized cells work together to form tissues. Different tissues combine to form organs. Different tissues combine to form organs, and organs work together as organ system. For example, muscle cells form muscle tissue, muscle tissues form muscle, and muscles function in the muscular system. Without cell specialization, complex body structures couldn't exist. Efficient functioning of the body. Specialized cells are adapted to perform their functions more effectively. Their unique structures allow them to carry out tasks faster and more efficiently. For example, the long axon of a nerve cell allows signals to travel long distances quickly. The biconcave shape of red blood cells allows efficient oxygen transport. Growth and development. Cell specialization is essential during growth and development. As organisms grow, cells differentiate to perform different tissues and organs, allowing the body to develop into a complex organism. Repair and replacement of cells. Specialized cells allow the body to replace damaged or worn out cells. For example, blood cells replace old blood cells. This maintains healthy tissues and organs. In a summary, cell division produces new cells for growth and reproduction, while cell specialization allows these cells to develop specific structures and functions. Together, these processes enable multicellular organisms to grow and function efficiently. Don't forget to like, share, and subscribe to Unlocked Science by Mishtab for more biology and chemistry lessons. Bye-bye.